Howdy everyone! Today we're going to talk about the balance of life in the ocean. We're also going to get to identify some phytoplankton and zooplankton species of Galveston. There's a balance of life in the ocean. Whether you're phytoplankton, a shark, a whale, or a small fish, everything has its specific role, this balance that each organism plays a key part in. We also, as humans, rely on the ocean for balance for food, because 16% of the world's animal protein comes from the ocean, whether that be fish or crab or shrimp. We all love to eat it. We also rely on the ocean for oxygen. Everyone take a deep breath in. And as we let it out, say, thank you, ocean. Because 72% of the world's oxygen comes from things in the ocean, like phytoplankton and marine plants. Also, some things that you can find in your own home or cupboard can be found or produced from the ocean. Kelp is in almost everything. Toothpaste, jello, ice cream. It's used as what's called an emulsifier, a thickening agent. It's also used in many labs around the world to grow bacteria for research on these plates called agar plates from the kelp agar. They also help with antibiotics. A lot of research on corals and sponges have been used to identify cures for bacterial infections such as pink eye and strep throat. So many things in the ocean we as humans rely on. To understand this balance, scientists will create food webs or food chains to understand the different interactions between animals. A food web consists of many food chains. A food chain is just one path an animal uses to find food. So when we look at food webs, there's a lot of intermixing of different species and different animals, squid and mackerel and marlin and tuna and small sharks and large sharks as well, all the way down to diatoms. But with food chain, we start with the energy from the sun and go all the way up to our consumers, our apex predators, our top predators of that food chain. The ocean food chain will always start with the sun, with our primary producers. Our primary producers are animals that produce their own food using photosynthesis, which helps an animal or plant turn the sun's energy into food, and they can create their own food through this process. And there are a lot of different animals that will use this process in the ocean, like algae or kelp, phytoplankton species, this random sea slug that will utilize algae in their body, just like zooxanthellae and corals. Zooxanthellae is an algae that will live in the tissue of corals and produce food by photosynthesis. We also have seagrasses and many more animals that I'm sure we don't even know about yet. The ocean food chain and the food webs will start with primary producers. And the most abundant being in our ocean is plankton. Now, plankton describes organisms that can't resist the current. A lot of people think of really tiny organisms that are plankton. But to use the term correctly, planktonic means anything that cannot resist the current, anything that's at the mercy of the current. So things that can get a little bit bigger are jellyfish. Jellyfish aren't very tiny. A lot of them are very large, but are as large as humans even. But these jellyfish are considered planktonic because they can't resist the water current. They can only steer which direction they want to go, but they actually cannot resist the current. It's like if you were to sit in the lazy river at Schlitterbahn. You're just at the mercy of the current, constantly going. You're not going against the current. There are two types of plankton. We have phytoplankton, which are plant-like plankton, those are our primary producers. They're the ones that use photosynthesis to be able to make their own food from the sun. A very common one is called Ceraceum. This green, you can see the green plant-like plankton will actually use bioluminescence, be able to light up to deter predators. So Ceraceum is a very common species that will be the base of our food chain, our primary producer. We also have zooplankton, which are our animal-like plankton, which are our consumers. These are like plankton from SpongeBob, if you guys remember. His name is Sheldon. He's an actual type of animal-like plankton called a copepod. So copepods and Serratium, those are types of plankton that are really small. You need a microscope to be able to see them, but they're everywhere. If you take a sip of seawater on accident, I bet you that there will be plankton in that gulp of water that they took, especially in Galveston, because we have very nutrient waters here in Galveston. So in food webs, the arrows will point to the animals that they're giving energy to. So as we follow along with food webs, the energy is going up that food web, up that food chain to the larger animals that it's giving energy to. No matter which path you take, the food chains and food webs always end up with decomposers, all right? So everything will always end up with decomposers, and a lot of that comes in the form of marine snow. All the way down to the bottom of the deep sea, down to the Marianas Trench, carcasses of animals will all go down in what we call marine snow. This is all the dead or decaying plant material or animal material or plankton that will fall down all the way to the bottom of the ocean, where they will be found by the decomposers. So there is a really wonderful balance from phytoplankton to sharks, from the top of the ocean where the phytoplankton are living to be able to produce photosynthesis, all the way down to the bottom of the deep sea. What we're going to do in just a moment is we're going to get to see a sample taken from Galveston Bay of phytoplankton and zooplankton species. What you guys are going to do is follow along with your guide, and you're going to be able to identify some of these species and maybe even sketch some of those species that you can see on the screen so you can remember them later.
We're going to go through and look at some examples of different types of phytoplankton that you may see in your sample. So again, you're primarily going to be seeing diatoms. You may every once in a while see a little round cell cruise across your slide, and that may be a dinoflagellate. They're typically much smaller, but those are mobile. They have the flagella. Diatoms don't. Diatoms do not have flagella, so they don't actually actively move. You will see them move in the water column, but it's based on lipid production in their cells. So they can actually produce more oils in their cells to make them more buoyant. So we kind of talked a little bit about density yesterday in class. So if they produce more oils in their cell, it's less dense in the water around it. It makes them more buoyant so they can float and stay in the surface, closer to the sun. Because they all have chloroplasts and they photosynthesize, all the phytoplankton do, just like grass or trees, how those are photosynthetic organisms, so are phytoplankton. So each of these cells, each individual cell has chloroplast, has a nucleus, and they function independently of each other. And you have photosynthesis taking place in each of these single cells, which is pretty cool. So diatoms alone make about 50% of the oxygen in our atmosphere, which is pretty cool. So you haven't thought about phytoplankton being important to you before? Now you know. So these are some of the common diatoms that we're going to be seeing. So Coscinodiscus is a really nice example. You should see a lot of these in your sample. They're disc-shaped. We're going to be looking primarily using your 10x objective, and you might go up to your 40x objective. On your worksheet, it asks you to put down what magnification you're looking at, or what you're viewing the specimen through. Whatever objective you're using, you're going to take that times 10, because the ocular that you're looking at is 10x. So you're going to take 10 times whatever objective you're using, and that's the total magnification. So looking at these to get this kind of a view, this was probably viewed using the 10x objective or 100 times magnification. They're pretty big. You'll see those even in the 4x, using the 4x objective, they're pretty big round discs. The Thalassiocyra, the individual cells are a bit smaller than the Coscinodiscus, and they also form chains. So the Coscinodiscus you'll see on their own, just single cells on their own. this is looking at, these are both Costodiscus, but looking at it from different viewpoints. So here you're looking at the side view of Costodiscus, and here you're looking at the top view. So they can look like totally different species, and you might just be looking at a different orientation of the cell. And that you just have to, you get better at recognizing that with practice. Unfortunately, that's about the only way to get better at that. The Thalassiocyra make chains. So there are these small discs, but then they have a little silica spine, or a little spicule, that connects the cells so they can actually form these chains. Here we're looking at Odontella. And again, these are both Odontella, but looking at different orientation, looking at different views. And they look totally different, right? So we have seen some Odontella, we saw some this morning. They have these 4 spines coming off of each side of the cell. Rhizoselenium, you may see some of those as well, very long and slender. And it also has these little spines that protrude from the ends. All of these little brownish yellow dots that you see, those are all individual chloroplasts. So you can have multiple chloroplasts per cell. And you have photosynthesis taking place within each of those chloroplasts. Why do you think that they have these little spines? What would be a benefit? No guesses. So they actually help provide increased surface area because the diatoms don't have flagella to keep them in the surface of the water column near the sunlight. So those spines actually increase their surface area and allow them to float in the surface near the sunlight a little bit easier. And they also change the lipid concentration or oil concentration in their cells to keep them more buoyant and keep them closer to the surface as well. This is my personal favorite. I love Chaetoceros. I couldn't tell you why, I just love it. I think it's beautiful. And each of these is a single cell. Each of these little rectangles is a single cell, and it has 4 of these long setae that come off of each cell. And these setae actually overlap with the setae on the cell next to it, and that creates this chain. And again, these long setae increase the surface area to help it stay buoyant in the water column. Here's another species, still the same genus of Chaetoceros, but it's more like spiral shaped. And so there's hundreds of species of Chaetoceros. We're only going to be identifying to the genus level. Trying to get down to species level is an art in and of itself. So we're only going to be going to the genus level, and that's what's indicated in the identification guide as well. Here you're looking at a single cell from the top down. So again, it looks very different. So you may think you found a totally different species or totally different genus, but it's actually still Chaetoceros. Here are some other pennates. So now these are diatoms in the class Pennaceae. The other ones we looked at so far were in the class Centraceae. So these are the pennates. So these are more elongated pennate shaped. And so navicula and pleurosigma are really easy to see in like your 10x, using your 10x objective, probably even using your 4x objective. These are really relatively long, large cells. Basilaria is really cool. So each of these little long rectangles is its own cell. And they actually produce this mucus layer. It's like a sticky substance layer that allows the cells to glide along each other. And so you can have this whole bunch of cells slide out along each other to make this long chain, or they can kind of come back together and form this more compact shape. But each cell is still functioning independently of the others. Just so super cool. Nitzschia, you'll see this maybe using your 40x objective. They're pretty small, maybe 15 microns in length, so they're pretty tiny. But you may see some of those, and these are all commonly found in our area. Here's a few more diatoms. So we have Thalassionema. And these, again, these rectangles, each of these cells is functioning independently, but they can connect by sticky substances here at the end. And they can form this kind of accordion shape, this fan-shaped structure, or you might have them make this kind of star-shaped structure. And again, with them linking together, it increases their surface area to help them stay more buoyant. Asterianeliopsis, each of these little triangles is a cell, and it has one long spine coming off of it. And then they make these chains as well. And again, those spines help keep it floating, right? Keep it more buoyant, increase the surface area. And with these and with Chaetoceros, with these big spines, if you have a big bloom of these, they can actually get clogged in fish gills and cause fish to die because they clog the gills of the fish and doesn't allow the oxygen to get absorbed into the bloodstream of the fish. So they don't produce a toxin, but they can still cause fish kills just in their sheer abundance. And this is called a mechanical means of causing a fish kill. So kind of interesting there. The lake morpho, we find that more when we have more fresh water. It has kind of this fan shape. You can see all the individual chloroplasts, multiple chloroplasts per cell. And so those are the most common diatoms that you'll be seeing. Today you might find others, and those are all present in your identification guide. So the dinoflagellates, some of the dinoflagellates can actually produce toxins. So I mentioned that when we were looking at the first slide with all the different phytoplankton up there. Some of the dinoflagellates can produce toxins that can either accumulate in shellfish or finfish, and then if we as humans consume those that have been exposed to that toxin, it can make us really sick. And if it's not treated properly, you can actually, in some cases, it can lead to death, but it's mainly due to the side effects of the toxin being present in your system. So it will cause like intense diarrhea, vomiting, so you actually can pass away by dehydration if you don't get treated. So the toxins, if you consume fish or shellfish that have been exposed to toxins, is really bad. So that's why we help provide that service for Galveston Bay to monitor for harmful algal bloom species, so that when we do see these species present, we can notify the health department or Texas Parks and Wildlife so they can shut down fishing in different areas of the bay wherever these species might be present or the toxins present. So this is showing a map of the Gulf of Mexico this is just looking at the different genera that are represented in this map. But the red areas indicate regions where Carina brevis is commonly found. Has anybody ever experienced red tide? Been exposed to red tide? Have you ever heard of it? No? So it's when this genus here, Carina, the species is brevis, when they're present in really high cell counts. So you may have thousands of cells per milliliter, even more. And they produce a toxin called brevetoxin. And this toxin can actually become aerosolized. So if you have these Carina in the surface producing this toxin, that toxin can attach to water molecules in the air. And when we breathe those water molecules in with the brevetoxin on them, it's really irritating to our respiratory tracts. So our eyes get really itchy, our nose gets really itchy, your throat might get scratchy. It might feel like you have really bad allergies. And so if you have asthma, this could affect you a little bit more severely than if you don't. So depending on your health conditions, you may be affected differently. But we do have Carina brevis present in our water sometimes in late summer months. We also sometimes have Dinophysis. This is Dinophysis ovum that also produces a toxin. And so these are the two that we're mainly looking for here because we know they've been present here before throughout different times of the year. So when we see these, we automatically notify the health department and get everybody kind of on high alert. They can go collect more samples, bring them to us, we run them for them, and then give them an idea of the cell count of these species across the bay. So we are watching. We're trying to keep everybody safe, right? We're going to jump over this. This is just looking at some little cartoon images of different types of dinoflagellates, and it gives their arrangement of plates. So they all have these little thecal plates. Some of the species you can't even identify by looking at them through a microscope. You have to look at them using electron microscopy to count these plates that are underneath the organic layer. So it's really, like I said, it's an art form to identify some of these down to species. So some of the common dinoflagellates that we'll see here, again, this is Dinophysis. It's also known as the evil princess because a lot of them are toxin producers, and they have this little crown up at the top, so it's called the evil princess. Not many of the phytoplankton have a common name, but that is one that has one. This is Serratium. This is a large, relatively large dinoflagellate. All these dinoflagellates have two flagella that help them move forward, or they spin the cell so they can make micro movements in the water column. I mentioned that the Asterianelliopsis and Chaetoceros with the long spines can get caught in fish gills. You can also have that occur if you have blooms of Serratium. With these long horns, they can actually get trapped in fish gills if you have a bloom of these species. Paradinium, this is a little bit smaller, so this scale bar is 20 microns, so it's about maybe 80 microns across. But you might see this swimming across your slide. If you see like a little green ball that's cruising across your slide, it might be Paradinium. We won't be able to identify it for sure using these microscopes, but it might be. It might be Paradinium. Acacia sanguina. So this is a dinoflagellate that we do have present here in Galveston Bay. And I did see this on one of our samples from a few days ago. So we may or may not see this. And again, it's pretty big. I don't have a scale bar on here, but if you're looking at this with the 4x objective or 10x objective, you would definitely see it. It's going to take up a pretty good percentage of your field of view using either of those objectives. Noctiluca. So this is the last one we're going to look at up here, and then we'll get to your slides. So Noctiluca is actually called mixotrophic. So it does not have its own chloroplasts, but it can steal chloroplasts from other phytoplankton. So it can eat other phytoplankton cells, digest all the tissues from those cells, but keep the chloroplast. And those chloroplasts still function, still photosynthesize when it's inside the Noctiluca. So they're called kleptoplasts. So they're actually mixotrophic because they do ingest other organisms for food, but they also can produce their own sugars by photosynthesis. So they're autotrophs and heterotrophs. Pretty cool. Another fun fact about Noctiluca, they're huge in terms of phytoplankton world. This could take up your whole field of view if you're looking with the 10x objective. It could take up your whole field of view. This is an example. They're about 500 microns across. If you're looking at a water sample with a lot of Noctiluca, you might see the little specks. So you can actually see the little cells. You can't see detail, but you can actually see the cells. They're so large, relatively speaking, in the plankton community. Another fun fact is that they bioluminesce. So this is a chemical reaction that takes place inside the cell, and a byproduct of this reaction emits light. So when you have blooms of Noctiluca, it actually makes the waves glow this blue, beautiful blue color. And we do have blooms of Noctiluca here along Galveston. I've seen it on the West End, I've seen over in Bolivar. Usually in the spring, it's usually foggy out, so it's like there's cool air, but the water's starting to warm up, so those conditions in the water make them happy.